Thanks, Lucas, for that very nice introduction. And well, without further ado, let's start. My talk is called "The Brain in a Vat," and we're going to be talking about constructing brains. Because while it is unlikely, it is still possible that you are just a brain created by some scientist or extracted by some scientist, put into a jar, and being fed impulses that represent everything you see, hear, sense, and experience as the outside world. This idea is not new. It's been with us for about a century in many different forms, in horror, in sci-fi hero's journey, in satire, or whatever sci-fi hero's journey trolling Elon Musk is engaged in these days. But I don't want to talk about the feeling of being a brain in a jar and how we might discern if we're one. What I would like us to concentrate on today is trying to get into the shoes of a scientist who might be trying to construct such a brain. We're going to talk about the motivations that the scientist might have, and we're going to talk about some methods that are being used today to achieve similar goals. But before we get to the how and the why, let's first talk briefly about the what. What being, obviously, the brain. So your brain, if you took it out from your skull, would look roughly like this. It's located, well, in your skull. It weighs about 1 to 1.5 kilos, and it does most, if not all, of your thinking. For many years, we thought, decades even, that since the brain is connected to the cerebrospinal column and to nerves that go throughout your body, that the nervous system, just like cardiovascular system that pumps your blood, nervous system is just a series of tubes, or like the internet. We've realized since that the brain is made of specialized cells called neurons, about 86 billion of those. And those neurons, I hope you can see my pointer, are made up of the cell body, the dendrites that span the space around the neuron and collect information and signals from other places in the nervous system. One long process called an axon, which we're going to treat as an output of a neuron, and then axon terminals that terminate in synapses later, which I'm going to talk about in a minute. And we know that partially because of the more than 100 years old drawings by Santiago Ramón y Cajal, who together with Camillo Golgi in 1906 got a Nobel Prize in Medicine for proving with those drawings that nervous system is, just like every other system in our body, comprised of cells. So neurons, as I said, are very specialized cells that are really good at relaying information very quickly over long distances. And the way they do it is with electricity. So here you've got an image of a cell body, and you can see this red black probe represented here with this little cone. We can see that if the neuron is stimulated enough, it will generate an impulse that we call a spike that will then propagate along the axon, passing by the blue probe and the red probe, and then terminating somewhere in the end at the synapses. And as you can see, the distances can be quite large for cell standards. And the times that it will take to propagate the signal from the very beginning to the very end are quite short. This signal then terminates at the, travels along the axon and then terminates at the synapse, where the electrical signal is getting converted into a chemical signal. The type of the neuron and type of the chemical that's getting released at the synapse is going to determine the effect of the neuron on the neuron that it interacts with. Mostly we're going to be talking about neurons being excitatory or inhibitory, depending on whether they make the next neuron more likely or less likely to fire, to spike. Well, it is very hard to actually measure these chemical signals that are happening all over the brain, it is fairly easy to measure the electrical activity of the brain, either by putting electrodes inside the tissue and recording the voltage of a single cell, or putting electrode arrays to measure the electrical activity of a certain area around those electrodes. We can also use the non-invasive methods like EEG, electroencephalography, or functional magnetic resonance imaging, where we can see the activity of the entire volume of the brain, either the electrical activity directly with EEG, sorry, this car is going outside the window, or the flow of the oxygenated blood in fMRI, which corresponds to the fact that neurons that fire use up energy, and they need to replenish that, and they need oxygen for that. So now that we briefly know what a brain is, it's a thing that does your thinking. It's an organ that is comprised of neurons, and those neurons operate on electricity mostly, electricity that we can measure. We can start talking about how we would go about simulating that. So you would be excused to think that, given how I started this talk, that what I'm talking about is actually creating an actual organ that you can put in a jar, pump it full of artificial cerebrospinal fluid, and keep it alive and try to understand what's going on there, and try to talk to it. While there is some research that is attempting to do something similar, they're creating brain organoids where they take mature cells from humans, they revert them back to the pluripotent or multipotent stages, and then they turn them into nervous cells so that they can grow three-dimensional structures that resemble parts of the brain that can be very useful. I am not going to be talking about that because, first of all, I'm not a biologist, so I wouldn't really know what I'm talking about. Second of all, this technique is still fairly new, so we don't really know how far we can push it. Since creating an actual organ is not going to be available for us easily, we can turn to the thing that scientists do when they are studying other systems of unimaginable complexity, like the universe, or the weather, or the stock market, in decreasing order of predictive power. We can turn to computer simulations. So how would we go about simulating a single neuron? Let's start with a single neuron. We can imagine a cell as a single point somewhere in space. Now, I said that neurons collect inputs from other regions of the brain and then produce outputs in forms of spikes. So obviously, we need to have some idea about what the input and output might be. So if we zoom in to that little dot that we just created, and we try to come up with a simple model of its activity, the simplest thing we can come up with is called leaky integrate and fire neuron, where if you look at the thing here, we've got a cell that's basically a bubble that's separated from the outside world via a double layer of the cell membrane, and it keeps a negative electric potential, negative voltage, with respect to the outside world. Since every neuron, like every single cell, is a very complex machine with lots of molecules and proteins that I know nothing about, we can do whatever scientists are trying to do in many of those cases. So try to express the thing that we don't understand and is very complex with an equivalent thing that is way simpler that we can describe. As you can see in here, we can represent, well, the voltage across the membrane. So the output that we're interested in would be spikes. The spikes happen when the voltage across the membrane reaches a certain threshold, as is shown in the plot on the right. The input that we get here is going to be the current that's coming into the cell. And in a setup like this, if a series of incoming spikes raises the voltage by enough in a rapid enough succession, we're going to see a spike, after which the cell will just go back to its resting potential, and then the whole process can start anew. This model is usable. It's used in many places, and it is very simple, so that's its advantage. But it's not complex enough for some people, and following the work done in the 1950s by Hodgkin and Huxley, who were awarded the Nobel Prize in 1963, they took axons from a squid. So it is a giant squid axon. And normally when people hear those three words, usually they remember it's an axon of a giant squid. I can assure you the squid was normal size. It's the axon that was giant. So if you want to ever have the satisfaction of correcting a neuroscientist about something in their domain, please remember this information because everybody gets that wrong. And they realized that we can represent the cell, the neuron, again as an electrical circuit. But this time, instead of just putting a single resistor for the leakage current, we can model the currents that are going through the membrane thanks to the flow of the sodium and potassium ions. And this flow, this current, is going to be mediated by the conductance, by the changing conductance of the ionic channels. That depends on time and voltage. All right, so we've got our voltage-based, current-based models, we've got our conductance-based models, we've got single neurons so far. But since we've got already this, we can try to connect them up. We can take a couple of neurons, say that some of them are inhibitory, so they are going to be lowering the voltage of the neurons that they're interacting with. Some of them are going to be excitatory, so they're going to be increasing the voltage of the neuron that they're interacting with. And voila, this way we've got a simple model of a brain. So what can we go, where can we go next? Well, we can go more complex. What you see here is a representation of the entire connectome, so all the neurons and all the connections between all the neurons, of a little nematode worm called C. elegans. That worm has 302 neurons, and we've counted them all. We know how they're connected, and we can try to model that. So how can we reach this level of complexity? Well, first of all, Hodgkin and Huxley said that we've got two types of ion channels, so the potassium and sodium. Turns out that you can make your cells as complicated as you want by adding different types of potassium channels, by adding some calcium channels, by adding some different mechanisms that are going to be adapting as the cell is getting excited and inhibited by other neurons. Then the possibilities are endless. Instead of imagining the neurons as point-like floating somewhere in space, we can actually embed them in three-dimensional physical space. We can say, okay, this neuron has got a body that's this size. It's got this many dendrites. They are located in space like this. And there are synapses that are affecting this neuron in those particular places. The plot on the right is from the lab where I work, from the Neuromuscular Systems Lab in University College Dublin, where a colleague of mine did a lot of work modeling really complex models of neurons. What else you can do? Well, you can add more cells. In our toy model, we had 6 of those. This paper contains about 1,000 cells. So if you think about that, we can go very close to actual biological description by just adding more complexity. We can add more cells. We can add more information about the shapes of those cells. We can add more mechanisms that are getting involved, but every single thing that we add is adding more complexity. And at some point, we actually need supercomputers to run the whole thing because this is just getting too computationally expensive. So there's another direction we can go with that, because not always we're interested in going as detailed as possible. I like this example that if you were given a map of a territory that was in 1-to-1 scale, that map is completely useless. It's a model of the world. Yes, it's a very accurate, perfectly accurate model of the world, but it's not going to give you any useful information. very tangible example, if you were trying to get from one place to another during rush hour in Warsaw, the map on the right would be much more useful because it shows you the metro stations and the connections that you can take, or you'll be able to take in a couple of years, than the map on the left, which is definitely way more complex and contains way more detail. But that detail, when you're asking a question about connections, is just noise. So another direction that we can go when we're trying to model the brain activity is to go simpler. First idea is to go to firing rate models. So if you look at the plot in the upper part of the screen, we can change all the information that's contained in the series of spikes that are present in the neuron and that are recorded from a neuron. We can replace that by a single variable that changes fairly slowly over time, which is the firing rate. So how many times per second that neuron is firing. This is adding some granularity in time so that all the computations that we have to do are way less expensive. Maybe we're not interested in that. Maybe we can actually encode some information just by the firing rate and not exactly by the precise timing of all the spikes. Another idea that we can explore in those firing rate models has been proposed by Wilson and Cohen in the 70s, where they said that, as you can see, this model looks very similar to the little toy example that I've drawn a couple of slides earlier, where we've had excitatory and inhibitory neurons. Here we've got excitatory and inhibitory neurons. But those are not single neurons, those are ensembles of neurons, groups of neurons that could contain tens, hundreds, maybe even thousands, depending on your needs, cells. As a result, we can definitely lower the complexity of the model and lower the computational cost by going way coarser in time and in space. Another avenue that we can take is to just forego talking about individual cells. Since we can measure the electric field, we can try to model the electric field directly and say, okay, fine, we can fit a time series here, and we can try to create a model that's going to generate something that's similar to this voltage if we let it run long enough. This is something that people are trying to do, for example, for the stock market, but in this case, we can actually predict something. Not in all cases, but in many more than in the stock market example. Another idea is to go even broader and say, okay, fine, so there are bits of the brain, and those bits of the brain oscillate. And we might not be interested in particular voltage. We might be just interested in the magnitude of the oscillations or the phase of the oscillations. Because, for example, if we're looking at synchrony, we'll be able to say that a patient's brain is sleeping because synchrony increases during sleep. Synchrony increases also during epileptic seizures. So this is something we might want to model. And this is called a Kuramoto model, where all those dots represent harmonic oscillators, which are like pendulums, but here represented in a phase space. So left to their own devices, they would just go around the circle with constant velocity all the time. But since they are coupled and they tug on each other with certain strengths, depending on the strength of this coupling, we might be able to see synchronization and desynchronization of the regions of the brain. So those are some ideas that people have been using here in real world, in academia, to actually describe brain activity and try to understand what's going on. Now that we've seen some of the methods, let's maybe ask why we would want to do that, because it's a question as important as any other. There are two broad categories of answers, of reasons, that I would like to present. First of all, are philosophical reasons. Second of all, is driven by medicine. As far as philosophy goes, those are really big questions that people have been trying to answer, because science has been in general very good at explaining things that we can touch, that we can measure, that we can experience in certain short amounts of time. So we understand why apples fall to the ground. We understand why the winds blow. We understand how the rivers flow. It might be, complicated in its own right. The technical details and the actual solutions might be eluding us for years to come. But we have the feeling that we understand the basics, and there's no grand mysteries in those domains, in things that we can actually touch and experience on a basis, on like everyday basis. All the big mysteries in science tend to be in the domains where we're talking about stuff that's very small, very large, and far away, or has happened a long time ago. Those are all articles from today from some popular science popularization websites. But at the same time, a human brain is something that you can literally just hold in your hands. It's tangible, everybody has one, and yet there's still so many big questions that we don't know the answers for. Like, how does consciousness arise? We don't know. Why do we sleep? We don't know. Where do we keep our memories? We don't know. What is a personality? We have absolutely no idea. And there's an argument that can be made that by creating thousands upon thousands of artificial brains and testing our hypothesis is going to give us more information than what we've been doing before we invented brain modeling and computer simulations, which was looking at people who had suffered brain injuries and figuring out what doesn't work when, for example, a steel bar, as in case of Phineas Gage, goes through your frontal lobes. In his case, his personality changed drastically. He used to be a nice and amicable man. He became very angry and very rude. He survived miraculously. The argument for taking advantage of thousands upon thousands of artificial brains is even stronger when we go to the domain of medicine, because there are still important questions about the brain that we can ask from a medical point of view. For example, we've got the neurodegenerative diseases like Alzheimer's and Parkinson's and Huntington's, which affect millions of people around the world today. And we still don't know what's the origin of those diseases, how to detect them early, and how to slow down their progression or even reverse it. And we would like to know those things. When we're designing things like prostheses, we've got some devices that work. You can see here a cochlear implant that restores hearing to people who are hard of hearing or deaf. You can see a brain stimulator for essential tremor or Parkinson's disease that works as well. But just because something works doesn't mean it works in an optimal way. We still need to find answers to questions like what's the best area to stimulate and what are the best stimulation parameters and how do we personalize the treatment, because something that works for one patient doesn't necessarily work for the other. So if we think about those questions and try to come up with the answers to those using the general framework of the scientific method, which is this gradual improvement process where we make observations, ask questions, formulate hypotheses, test those hypotheses, and then update our model about the world, and then make more observations. While we need to take everything to the clinic, everything in medicine needs to be tested clinically with patients repeatedly. What modeling does, what computational modeling does, is lets us greatly speed up this little subloop in the process. Because while we won't be able to find all the good solutions just from the models, because all models are going to be defective in some way. They're not going to be able to capture everything. But if we find something that we thought might be a solution and it doesn't work in the model, we might be fairly sure that it won't work in real life either. This is basically going down the same route to invention as Thomas Edison, who once said that he has gotten a lot of results because he knows several thousand things that won't work. And that's a very valuable lesson. For a concrete example, I'm going to briefly talk about the stuff that I do at work, where we try to design new and improved methods of stimulating the brain in Parkinson's disease. So the way it's being done clinically these days, and we've done that for about 30 years, tens of thousands of people worldwide are taking advantage of this treatment. You put an electrode and a pulse generator inside the patient so that the electrodes reach deep structures in the brain, and you stimulate them electrically and it improves the symptom of Parkinson's disease, lets the patients lower the medication dose that they're taking to limit the medication-induced side effects. But obviously, a system like that is not without its own problems. There are stimulation-induced side effects as well. The needs of the patient might be changing throughout the day, throughout weeks, months, years as the disease progresses. And ideally, we would like the stimulator to adapt to that. So what we're trying to do, well, not only us, but the framework that we're working in, is where the stimulator doesn't just send the stimulation without regards for behavior and the brain activity. We're trying to measure the brain activity at the same time. And based on what we measure, we adjust the parameters of the stimulation. So what we've done with my collaborators a couple of years ago is we proposed a self-tuning controller where some of those parameters would be adapted in time. And then we tested that on 2 different models of the same structure of the brain. So one of them was a Hodgkin-Huxley type conductance-based model, very intensive. It had to run on a supercomputer, and it would still take like a day to run all the simulations. And then we had a simple firing rate model that we could analyze mathematically, which we couldn't do with a conductance-based model, and draw some conclusions. And I could run the simulations on my laptop within 15 minutes. So having seen that the controller that we propose outperforms classically used controllers, we were able to suggest that it's going to work even better if we move it to the patients. Like right now, we are working on that. We've created a platform where we can test things in a closed loop in animal models, not in patients yet. And we'll be testing those advanced self-adapting controllers fairly soon. And we were given that confidence by the fact that we've actually seen it work in multiple different scenarios that we've thrown at it. So if you go back to this whole idea of modeling as a tool in scientific method, sure, we might not be able to find this optimal stimulation that way. This is still going to need clinical testing, and we will still need to work with physicians, with patients, and it's still going to take a lot of time. But we've been working on that for about 10 years. Most of the clinical testing with patients has been done for very short periods of time, which is not ideal, on small groups of patients, which is also not ideal, in post-operative conditions. And sometimes those patients were tethered to a computer, not able to live their lives with the device. So by finding something that seems to perform better and rejecting a lot of ideas that we definitely know are worse, we will be able, that's our hope, to get everybody to focus the efforts so that we can get everything that we propose tested clinically and get it approved quickly so that we can improve people's lives. Sure, this is going to be a slow process. It's going to take a lot of time, but we are going to be chipping away at all the problems that come out of the clinic, trying to solve them bit by bit. And who knows? Maybe we might even find some hints pointing us towards the answers to the questions about consciousness and all the other stuff. So that is all from me. Thank you very much for your attention.